The assay that I chose to look at for my final project is an ELISA. So, what is an ELISA? This is an enzyme-linked immunosorbent assay, and it's used to determine the presence or to quantify amounts of proteins in a sample. And to do this, it's going to use antibodies that detect the antibodies, antigens, and proteins that we want to see. And if you're unfamiliar with these terms, an antibody is a protein in our immune system that can bind to specific proteins. And these specific proteins are called antigens, and they're found on the surface of cells. And then this diagram down at the bottom here just shows the most basic steps of an ELISA. So this first well here has the molecule that we're looking to detect in green. And then the second well is when you add the antibody. And this antibody is the orange and pink Y-shaped thing here. It's going to bind to the protein that we want to detect. And in the third well, you add a substrate, and this substrate is going to react with the enzyme that is attached to the antibody to cause a color change reaction. And that's how we'll know if the protein we're looking for is present in our sample. There's a few different types of ELISAs. Firstly, there's a direct ELISA, and this is going to quantify the amount of protein in a fluid sample. Down here, you have the primary antibody conjugate, which sounds like a complicated thing, but it's really just the antibody that has the enzyme attached. And when this antibody binds to the protein that we're looking for, and a substrate is added to the sample, it's going to cause that color change reaction shown on the last slide. An indirect ELISA is almost the same thing. It also quantifies the amount of something in a sample, but it's going to quantify antibodies rather than just proteins and antigens. And to do this, there has to be a secondary antibody conjugate, which is where the enzyme is attached and the color change reaction is going to occur because the primary antibody is the molecule that we're actually looking to quantify. And then a lateral flow ELISA is something that you're probably familiar with, even if you don't realize it. This is what a COVID test is. So it just determines if the protein is present in a sample. And to do that, it has the test line here and the control line here. So the sample that we're looking for is going to be added, and then an antibody will bind to it if it's present. And that antibody is going to also bind here at the test line, and that's what causes that line to appear if you do have COVID. And then the control line here is always going to be seen if the test is working properly because another substrate will always bind there and that color will always be present. So the specific paper I looked at was called Sex-Specific Associations Between Cartilage Structure and Metabolism at Rest and Following Walking and Drop Landing. And in this study, they used a direct ELISA to detect cartilage oligomeric matrix protein, abbreviated as COMP. And this is an antigen that is released when our cartilage is degraded. Down here in this first well, you see a little red circle, and this is representing COMP. And normally there would be a lot more in a sample, but there's only one here to help you visualize it easy. Oops. And once you add your fluid sample to the well, the protein that you're looking for is going to attach itself to the plastic edge of the well, which you see in step 2, the little red is down at the bottom. And then in step 2, you're also going to add your antibody conjugate, and this is just the enzyme that has the antibody that has the enzyme linked to it. And if your protein is present, which is COMP here, and you can see it is there, the antibody is going to bind to it, which we see. And then in the third step, you add the substrate that reacts with the enzyme. So you add this TMB substrate, and it reacts with the enzyme to create the orange-colored product. And the amount of orange-colored product is what's going to tell you how much cartilage oligomeric matrix protein is present in the sample, because it's directly proportional to the protein. So yeah, it's actually fairly simple. That is how these researchers were able to detect the level of cartilage oligomeric matrix protein in the sample. And these are the references that I used. Thank you for listening.